All right, and
You weren't supposed to mention.
Right.
Sounds me.
Professor Hilda, take one. I'll be clearing these lights. Can you see?
Hmm?
And clear.
Hi there.
Hi. Hi. How are you?
Tired and grumpy. I thought I'd just ask asking where did you go to school and how did you get into science? What prompted you to go into science?
I went to school in England, in a town called Hitchin, and I really liked biology at school. I was definitely a biology nerd. I think what really promoted me to go into science was my real interest in understanding why you inherit some things from your mum and some things from your dad, and the whole idea of genetics and how genes control everything in your cells. And that really is what promoted me to start working on human DNA.
Was there something distinct about your mother and father you noticed in yourself?
Oh, for sure. I'm not sure if they're good things or bad things. People say I walk like my dad. I'm not sure about that. I think it's also something very relevant is how different I am from my brother, but similarly we share 50% of our DNA. But we have a lot of similarities too. And I was always fascinated about that kind of mix of traits you share with your siblings.
So you finished high school and then you thought science was a good first choice, science or biology? Or?
Yes, biology. I did biology. That was what I was interested in. I went to Birmingham University and I studied a lot of plant genetics there, but I really wanted to get more into human work. So for my PhD, I moved to Leicester University and started working on human DNA there. Yeah.
How long did that take?
My PhD I studied for 3.5 years and wrote my thesis.
Tell us about your thesis. What was your thesis?
My thesis was on the molecular characterization of telomeric DNA. So,
We can unpack that.
yeah. Yeah.
And what year was that? So telomeres are recent discoveries, aren't they, or not?
Relatively recent. But I started my PhD in 1998 and I finished it in 2002. Yes, I did 4 years of work understanding telomere sequences, telomere DNA, and how they work to regulate cell growth.
So which path to take? No, let's try and pack down telomeres. Do you think you can summarize them in a sentence?
So telomeres are the caps on the ends of the chromosomes. The common analogy used is the little caps on the ends of your shoelaces, the little plastic pieces that hold your shoelace together. So your telomeres function in a very similar way by capping the ends of your chromosomes and preventing them from becoming unstable and degrading.
Okay, so, excuse me, thank you for that. Excuse me, Andrew, I'm going the right way, far too much. So you went into research though?
Yes.
Yes. Tell me why.
My dad is a researcher, and I was always exposed to going to the lab with him at weekends and sitting while he did experiments and playing with the pipettes. So I was exposed to research all my life. And I really like a lot of aspects of a research career. It's a constant change. There's new developments in technology. You're constantly learning. Every day is a bit different. It's exciting. Sometimes it's quite heartbreaking, but the excitement, I think, outweighs anything else.
So how did you start? Your first research path, okay, so you've done your PhD and then now what?
Yeah, so I did my PhD, and that was very much based on looking at DNA and understanding DNA in cells. And what I wanted to do then was to really explore what the DNA does. So the DNA encodes proteins, and so the genes encode proteins which do the action jobs in the cells. So what I wanted to do after my PhD was to really understand a little bit more about gene expression and the proteins that are made. So for that reason, I pursued a postdoc position, which is a position after a PhD, for Cancer Research UK in Leeds in England, where I started working on a tumour suppressor gene involved in bladder cancer. And I did that for 3 years prior to moving to Australia.
So I'm going to break that down.
Yeah.
Tumour suppressor.
Yes.
Is that what it sounds like?
It's a protein.
That takes,
yes.
Yeah.
It stops.
Suppresses a tumour.
It suppresses the tumour growth. It suppresses the growth of the tumour cell. So,
Could
yeah.
you just say that? So?
So the tumor suppressor gene suppresses the growth of tumors. It stops the cells becoming cancerous.
So you really hit the ground flying with research. You weren't intrigued by medicine or anything else, or straight into...
No, I like research. I like working with cells and DNA, maybe more than people. But and I think there's a lot of very exciting things you can find at the molecular level that I think is maybe not what a clinical path would take me on.
Okay, I'm going to ask you to help us out with something. One of the plans we've got within this documentary, did you ever see The Big Short?
No.
Okay. It's a great film about the economic collapse in America in 2008, about shorting the entire market. It's highly technical when it comes to economics, and highly technical if you don't know the words in the financial. So the way they got around that was, if there was something that was really difficult to explain, they cut to, in one case, Margot Robbie in a bubble bath with a glass of champagne explaining what that meant. Another time, Anthony Bourdain using a metaphor of cutting up fish that stay old for. So we'll be doing the same, but if I can get it in really distinct ways, I'd like that to explain proteins. And I know I'm hitting you out of left field with that. You can think about it if you like, but it's like, to understand everything, I think we need to understand what proteins are to make sense of what you're saying. Or am I wrong?
No, I think you're right. I think
It's okay, you don't need to answer it now from the way.
no. I'm not sure of the best analogy, but I think DNA is like a big book of words, and I think each gene is like a sentence in that book. So although you've got a lot of words and a lot of letters, you actually need to make meaning of that. And I think sentences are the meaning. They mean what you need, what the words mean. But then I guess proteins are kind of the action part. So they're the bits that do the next step. So maybe, what would that be?
Would there be letters? No.
No, the letters of the DNA. The letters of the DNA. The sentences are the genes, because that's a segment of the DNA. But I guess the protein is the action part. It's what's made from these letters. So the action of doing what's in a sentence, or a book chapter, or something that's made for that. I don't know. I can't think of a better thing.
No, that's fine. And I recognise I put you on the spot. Don't hate me for it.
I'm trying to think of something else.
Maybe the story.
The story. Yeah.
Maybe
Well, you can go story, chapter,
maybe the
sentence,
words.
paragraph,
So the-
letters.
So the proteins of the story, is that it?
Yeah.
Is that the action things?
The action.
Yeah.
Yeah.
She's
I'm trying to
going,
think of something
whatever.
better now. I'm trying to think of something
No,
better.
say that again, is that the- no?
Trying to think of something better.
Well, you're not going to think about when you're on the spot.
No,
like
I
that,
don't think I'm
is
going to
that-
manage this. That was going to come to me later, isn't it? I'll be like,
in
that
the
would have been
shower.
great. Yeah.
Okay, well, no, something might pop
Yeah,
up
yeah.
as we go forward. So you didn't have any great vision about, you've always wanted to be a researcher?
Not always. I've faltered along the ways, as everyone does. I think science is a hard career. You're stifled by funding. A lot of it's based on your track record, your publications, and that's, you need that to get funding. And so when you're not publishing, you haven't got any funding, and funding is really hard. Currently the funding rate for NHMRC is 10% of grants get funded, so there's a lot of disheartening times. Also, experimentally, when you're working in the lab, a lot of things don't work or produce different results, or simply experiments just don't work, and then you have to go back to the drawing board and try again and figure out why it went wrong, what went wrong in your experiment, why didn't it go to plan, and why didn't it work. So there's a lot of times that I think you need to be quite strong to get through bad times to keep going. But yeah, I've always really, really enjoyed science. And I think when you do have a success and something works out and you find something new, that those kinds of moments take a lot to beat, I'd say.
Well, someone did say being a researcher was a bit like being Sherlock Holmes
Right.
in trying to decipher or look it out. look at problems.
Yep.
What was the famous quote? You know, once we removed everything that's impossible, whatever's normal, no matter what's left, no matter how improbable, is probably the answer.
Yep.
Have you
Yep.
been surprised by many results in your life? And well, when you say experiments fail, is fail the right word?
Well, I think there's two aspects. There's an experiment can fail because you forgot to put something in it, or something didn't work, or something went wrong. But then you can have experiments that don't go quite how you wanted them to, or how you expected them to, maybe more so. And that's what's really interesting, because when something doesn't go quite how you thought it was going to go, but it has worked, that's when you can find some really interesting things out, I think.
Do you have an example? Has that happened to you?
Oh, so I mean, certainly one gene we were working on, we thought it would do something in some cells, and it did kind of the opposite. But actually, that turned out to be super interesting, and we turned out to be able to kill cancer cells because of it. So, yep.
You better expand on that as much as you can.
So do you want me to talk about the gene in particular? Can I mention
Well,
gene?
Well, the throwaway line and happily called cancer cells is like, yeah, okay. We might go a little bit deeper into that.
So we identified a protein encoded from a gene in cells, and we were interested in looking at what it did in a particular pathway in the cells that we were looking at. And we thought that it would enhance the pathway that was happening in the cancer cells, and it did. But what we actually found at the same time was that this turned out to be toxic for the cells. So we were making the pathway go to such an extreme that the cells really couldn't cope with this particular process happening. So they shut down and died, and that's what we were really interested in, and we didn't see that coming on the horizon initially.
When you say, and it's just look, it seems very attractive, but you can look at me.
Sorry, yeah, not very good with eye contact. I'm a scientist.
Pathways. When you say pathways, just expand a bit on that.
So pathways,
just go back and talk
yeah.
me through it, because one, I'm finding it really interesting.
Yep.
So, and I really want to understand
Yeah.
what you did and what happened.
Yeah.
And there must be an almighty eureka or oh.
Yeah. So in the cells, the genes encode proteins, and the proteins do the job in the cells. But the proteins don't work in isolation. You have a lot of proteins that work together, and they do different things in different stages. So, for instance, I'm particularly interested in DNA repair pathways, so how the DNA is repaired each time the cell divides, if there's some mistakes that are introduced. And so the proteins that actually work in this way to repair the DNA problems, they don't work in isolation. There's multiple proteins that come in and deal with different types of damage, and they pass the sort of buck along down the pathways of the proteins to identify the error, and then to bring in other proteins that then correct the error in different ways. So the whole network of genes and proteins in a cell is huge and vast, and you need to try and understand a lot of knock-on effects and how these proteins interact with each other to understand what's happening in cells as they grow and as they become cancerous.
I'm going to ask you just to slow down a little bit.
Yeah, that's a common.
I admire it. I think you're dedicated, but I'd like to do that again, because I'm sort of there, whether the audience is sort of there or not, but I think you're explaining it really well. Just take your time and walk us through that, and then how it related to you after your finding. Because you said it didn't go to plan, but it was like, oh, well, we found this. So that's one of those moments in research where you find something you're not expecting, right?
Yes. Yes. So we identified that a protein that when we depleted it in cancer cells caused the cells to die. But what we were expecting was that when we got rid of this protein, it's not a good thing to say, is it? I don't think that came out right.
Following you.
Yeah. Maybe I should start.
So, excuse me, I don't know what's wrong. Okay, let's just break it down step by step. So you had a hypothesis?
Yes.
Okay, what was your hypothesis?
So we were hypothesizing that when we depleted the protein or inhibited the protein, the cancer cells would keep dividing, and they would become more active in a particular pathway or process in the cells.
So you were depleting?
Yes.
Well, taking it away?
Taking it away.
Killing it?
Inhibiting a protein.
So
Inhibiting
how
a
do you
protein.
inhibit a protein?
So we can either use a drug, but in this case we were using genetic depletion. So we were using a short interfering RNA, which is able to get rid of the protein in the cell.
Okay, let's join all that together now that you've explained it, and you're doing great.
Right.
So start from the beginning, but when you say deplete, explain
Okay.
what it means, because then we'll understand what you're doing.
Right. So we were genetically getting rid of a protein by knocking it down using a technology called siRNA, which causes the protein to be depleted in the cells. And what we did was when we depleted this particular protein, we were expecting the cancer cells to grow more because we were activating a pathway more. But what we actually found was that when we did this, we caused so much activity of this pathway that it was actually toxic for the cells. So the cells died. And that's what we're really interested in, trying to actually take on board this discovery and actually use it in our favor to kill cancer cells.
So why did the cells die?
Ultimately, they died.
Sorry, just go. I'll clear my throat and then you can start again.
Ultimately, the way it was working is that the cells were having too much damage in them, too much damage to the DNA. So the protein we were interested in was a DNA repair protein. And when it didn't work, we were getting so much damage to the DNA that the cell shut down. And so all our cells in our body have mechanisms to identify when there's damage in the DNA, because too much damage is dangerous because it can cause a lot of mutations. So we were interested in trying to get more damage. Oh, that's not right.
We were interested in getting.
So
Yeah, that was good. I was following it all.
yeah. So when we depleted this protein, we got more damage, but it was too much for the cells. It was too toxic, so they shut down and died.
And so all cells were killed.
All the cancer cells that use this particular pathway. Yeah.
Do other cells apart from cancer use that pathway?
No, that's the great thing. So this pathway is specific to cancer cells. It's active in about 5 to 10% of all types of cancer cells, but it's activated much higher prevalence in some bone and soft tissue sarcomas in particular that we're interested in treating.
So what do you do with that piece of information?
So now what we're trying to do is take that discovery and taking it further by developing drugs that can actually inhibit this protein. So chemically inhibiting it so that we can administer drugs to patients and that the cancer cells will very selectively die.
So you're targeting them,
Yes.
and that's what the exploration of the drug is going to be?
Yes. So the idea is to firstly develop drugs. So there are no drugs for this protein at the moment. So what we're trying to do is to actually take the structure of the protein, but also use high throughput screening approaches to try and develop drugs that very specifically target this protein. And that the idea is then that these drugs will be able to kill very selectively these cancers.
These cancers typically, I understand that cancers are different for different people, even if they might occur in the same areas, but are these cancer cells typical of anything in particular?
So this pathway is active in about 5 to 15% of all cancers, but of all cancers, yeah. But the prevalence becomes much higher in some bone and soft tissue sarcomas, particularly leiomyosarcoma, also pancreatic neuroendocrine tumors, osteosarcomas have a very high prevalence. So about 60% of some of these cancers activate this pathway. And these are the types of cancers that we're particularly interested in trying to treat with the therapeutics that we're developing. And a lot of these cancers are very rare, they're hard to treat, the prognosis is very poor, and in many cases, the treatment regimes haven't changed for a really long time. So there's a really big need to try and develop new personalized and precision medicine treatments to try and actually combat these cancers.
Well, pancreatic cancer is a bitch, isn't it?
Yeah.
It's a real killer.
Yeah.
Why is that? What's so virulent about the pancreatic cancer?
So I don't think we really fully know exactly why some cancers are so much more aggressive than others, but there's a lot of genetic changes that happen in these cancers, and that's what we're trying to understand to try and make treatments much more personalized and much more targeted and much more selective.
When you say genetic changes,
Yes.
just talk me through that.
So changes to the DNA.
Slow down a little bit.
Yeah.
So I know you're anxious to catch the bus.
I just talk like this all the time, sorry. So cancer cells typically have undergone a lot of genetic changes. So this means changes to the DNA. So as I was saying, every time your cell divides, your DNA must be copied to each of the daughter cells. But every time your DNA is copied, errors can be introduced in the DNA. So the cells have a lot of proofreading mechanisms and repair mechanisms that correct these processes. But what happens in cancer cells is these changes can accumulate, and these can essentially lead the cell to not realize it needs to stop dividing. All the barriers that normally stop a healthy cell from dividing become overridden. So cancer cells continue dividing and they accumulate damage, and this essentially promotes them to keep growing.
Could you simplify that even further? I felt no, you're just going, well, you kill them.
Yeah.
But even not, that's a little bit more complex than that. But
So cancer cells go through a clonal expansion. So the DNA, the cells keep replicating. And so essentially that's what a cancer cell is. It's a cell that doesn't know when it should stop dividing. It's a cell that just keeps going. And in this time that the cell keeps dividing over and over again, it goes through multiple divisions, and errors can arise in the DNA, which cause the cells to keep becoming more and more unstable and growing further.
okay, great. Did you buy that? I bought it. It's like dropping a rock into a pond.
I don't think it was incorrect.
No, I do find it fascinating. Is that your Hail Mary moment, as they say, the Holy Grail?
Oh, when we identified this
Yeah.
protein.
And how do you do it? I mean, well, physically, what do? You look into microscopes. You put petri dishes out. How do you do this?
So it is looking at in petri dishes and microscopes. You know, we work on human cells almost exclusively, so human cells in culture. So we grow them in flasks or dishes, and then we can look at them, we can do things to them, we can knock down genes, we can overexpress genes, we can put tags on genes so we can see them in the cells, we can see where the proteins go. So these are the kinds of experiments we use. It's molecular biology, and that allows us to identify things in cells.
Lost your way at the end?
Yeah, I did a little bit, yeah.
We'll come back. Try that again if you want to. Well, look, we might even show somebody doing it because
Yeah.
the big thing, visually, people want, well, okay, the reason, how does it work? I mean, it's
Yep.
is it in my head, and it probably is exactly what's in their head.
Yeah.
It's a petri dish and a pipette. And
Yes, absolutely.
But it's like,
Yeah.
no one's ever done it. But,
Yeah.
and no one sees it except in the most generic of ways. So it'd be nice to have them understand a little bit
Yes.
of how it's
Yeah.
done, if you don't mind.
Oh, you want me to say that all again?
Yeah, but
Yep.
slower and
So the type of work that we do is looking at cells, looking at human cells. We grow them in culture, so they grow in dishes or plates. And this allows us to manipulate these cells really, really easily. So we can knock down genes, we can overexpress genes, we can put in drugs, we can put in tags so we can look at proteins and where they localize in the cell. We do a lot of work with protein extraction and DNA, running them out. Oh, that's not really good. Protein extraction and DNA. We do a lot of microscopy work. We do a lot of live cell imaging where we can actually track cells, particularly cancer cells, and see how they divide and see which stages of the cell cycle when the cells go through to divide each time, where the problems arise and what happens to the cells.
your greatest surprises when tracking these sorts of things?
I think one of the greatest things is not looking for a specific thing, because cells are really fascinating to look at when they divide. And, you know, there's a lot of abnormalities that happen, particularly in cancer cells. So it's always a little bit of a surprise to see some of the things cancer cells do to keep dividing. They're really very aggressive, some of them.
Oh, okay. Do you have a vision of those?
Oh, so some of the cells can have little micronuclei forming that spin off, and there's DNA in these structures, and they can form very big nuclei, and there's a lot of abnormal developments that can happen to cells as they try and divide. You can get asymmetric splitting of the cell, you can get extra fragments forming, the cells can bleb. It's
Bleb.
Yes.
Come
Bleb.
on, bleb. What's bleb?
Bleb is kind of forming a little bleb. Yeah,
Yeah. Okay.
yeah,
I'd love to see a vision of that.
yeah.
Can we film?
Yeah, you should be able to fit. We have live cell videos and stuff. So
Fantastic.
Yeah.
Okay, well, that'll
Yeah.
help.
Yeah,
Too.
yeah.
We can show them what
yeah, and we have some really nice cell tracking systems as well, where the cell is red in one stage of the cell cycle and green in another stage of the cell cycle, so we can see just by a color which stage of the cell cycle it is in. So that's really nice to visualize.
Yeah. Okay, so we established that there was no particular influence for you wanting to do research except your interest in biology.
Yeah, I just loved biology when I was at school. I've always really found biology fascinating, and yeah.
But maybe your dad had something to do with the research.
He's a chemist.
Do you look down upon chemists?
No, I don't. I have an awe for chemists. I actually work with a lot of chemists now in terms of a lot of the drug design that we're using. You know, I have a lot of collaborations with chemists throughout the world, and you know, I think that's a huge part, and that's a huge part of science, is collaborating with different scientists. It's very multidisciplinary. I'm a molecular biologist, but I work with bioinformaticians, with biochemists, structural biologists, chemists, and this really maximizes what we can get out of research.
Well, some people say that science is jealous and people can be quite jealous of their turf. Has that been your experience?
There's definitely an element of that, but I think ultimately science is incredibly collaborative, and I think that's one of the beauties of science. Mostly scientists are more than willing to tell you all about their science and to work with you. Scientists really, really, really do enjoy that. Yes.
Talking to people who understand what they're doing.
Yeah, absolutely. Or not, and explaining what they do, or trying.
Which brings us here.
Yes.
I don't know where else. Well, what else would you like to tell me about what you do?
Oh, you asked whether it was a sort of eureka moment. Yeah, it definitely wasn't a eureka moment. You know, we these discoveries took a long, long time, many, many years, and it definitely wasn't a specific situation where we were like, wow, we've really found a target here. It took a long time to kind of realize the implications of what we had discovered, and working together to think about that and how we could use it and what the next steps would be as well. Because making a discovery in the lab, you really need to then apply it. So you need to then decide what the next steps are. How are we going to try and drug this protein? How are we going to try and make a compound that inhibits it? And what will the stages after that be to take it forward to the clinic eventually?
So it's, well, one of the things you said was going to be good for a soft tissue.
Yes.
And that's finding cures for soft tissue is particularly difficult.
Finding cures for some of the cancers that this pathway is very active in is a really big priority. They've been very recalcitrant to treatments so far, and there's not really any good treatment options that are very targeted. So there is a lot of need to develop new, improved technology and treatments.
Has this discovery led to other discoveries or other thoughts in your mind?
It's definitely led to a lot more informed choices. So understanding the pathway to take a discovery in the lab forward to potentially to the clinic. So a lot more awareness of intellectual property, of the process of how you develop drug screens, how you engage clinicians and chemists, and how you take things forward towards the clinic. So that's something I really have like to bring to other projects in my lab, and something I really want to take forward as a kind of pathway to take discoveries that we make forward.
Well, other projects, how many projects are you running at any one time?
Maybe 10 projects in the lab at the moment, I'd say.
Rough ideas of what you're looking at?
We've got some projects looking at telomere length maintenance. We've got projects looking at cell cycle regulation, combination therapies for glioblastoma. We're also looking at some projects looking at activation of telomere maintenance mechanisms.
Okay, let's go to telomeres.
Yes.
You want to explain that in a sentence?
So, what a telomere
It's
is.
making you more beautiful.
So we're very interested in.
Wait through. And look, this is tricky. I know this is tricky. So
What are we talking about? Telomeres and alt.
yeah, well, use of the length.
Yeah.
That's one of the projects. So
Yeah.
I'm trying to.
Yeah.
Well, I want people to know that you're juggling.
Yeah.
Well, not juggling, but you've got a lot happening at any
Yep.
one time. So it's going down more than one path.
Yes.
And then to explain just a couple of paths so people can demonstrably see that they're different, although joined in some way, but different. So you've got
Yep.
a broad vision, and that's kind of interesting because you're going down many paths at once, or maybe you're not. Maybe it's the same paths with different, same ending but different paths to get there. I
Yep.
don't know.
Yep.
And this is what I'm hoping you can explain to me because I'd like to make this sound as exciting as I think it is.
Okay. Right.
So no pressure.
Okay.
Yes, whenever you
Oh, I'm ready. You're ready. Okay. So my lab works a lot on telomeres, and telomeres are repetitive pieces of DNA at the ends of the chromosomes. The common analogy is the plastic caps on the ends of the chromosomes. Telomeres are fascinating because they shorten every time your cell divides, and this is because they're at the ends of the chromosomes, and your DNA can't replicate to the ends of the chromosomes. So there's a problem, and they gradually shorten each time your cell divides. But what happens in cancer cells is that these regions become extended, and that's what really allows cancer cells to be able to divide indefinitely. So they need to maintain these structures that regulate cell growth and cell proliferation. So one of the things that my lab is interested in understanding is how these pieces of DNA, these telomeres, become extended in cancer cells. Because essentially, if we can block that process, we have the potential to be able to stop cancer cells from dividing. So this is a big area of research in my lab, but also we're interested in the aging aspects of these telomeres. So as your cells divide, these telomeres get shorter. So this is essentially part of the aging process. So we're interested in understanding telomere biology from the context of cancer, but also in the context of aging.
I thought it was good. Did you think it was good? Did you understand it?
Yeah, I got it, yeah. Just about.
You didn't do anything about the history, well, when you were studying, you didn't do any of the history of research at all, did you? It was just like, no. So those questions are out.
I was trying to think, I was trying to remember when you were asking me earlier the exact date that telomeres were discovered and when 1994 was it? Yeah, I was like, God, I can't remember that. That's weird.
Anything else you want to tell me? Excitement, failures, depression, followed by overwhelming joy.
There's definitely a bit of overwhelming joy when you make a discovery. I think science is riddled with disappointments as well as successes. I think something that is often maybe not understood is the need to publish. So we publish all our discoveries, and sort of publishing work is a hard slog as well. There's a lot of data required for a publication. All publications in good journals go through peer review, so they're reviewed by our peers. And I review a lot of manuscripts for other people, but the peer review process can often be quite arduous to get the standard of the research really to a point that it's unequivocal what you're finding.
So is peer review like your English teacher marking your essay, or is it in more depth than that?
I'd say it's in more depth than that, maybe. So typically an article would be reviewed by 3 or 4 reviewers from different places, different seniority, and they will ask for rigorous experimental clarification, textual clarification to make sure that it's really understandable, to make sure that the level of the science is really robust.
Do you have any questions? I know you've inquisitive mind.
No, I think that was
Okay.
You've made him boisterous. Yes,
Yeah.
Andrew, of course, you're amongst friends.
when you just talking to Stuart, when you design a drug that will attack the cancer cells that you're trying to attack, how much research goes into after that event for possible side effects of the drugs until it gets released?
Yeah, so that's a really interesting question. So the work we've done, we discovered this protein involved in this particular pathway in cancer cells in 2019. We have established a company to develop drugs against this target. I also have academic collaborations that I've had for over 10 years trying to develop drugs for this particular target. We have some compounds at the moment, but then the next step is to test them robustly in preclinical tests, in cell lines and in spheroids, but then also move into animal models. So the idea is to test them in rodents before we go into humans. And then you need to design clinical trials. So it can take, 10 years, 15 years, 20 years to do these things. It takes a very long time. We've been working on it now for many years, and there's a long way to go.
But COVID was very quick.
COVID was very quick because it had to be. So, and also a lot of the processes in place with the virus and the viral technology, with the, what's it called?
That word, that word.
Yeah. With the vaccine, the process for the mRNA vaccine had taken many, many years of research to get to that point that it could then be applied for COVID. So all the research had been in place for many, many years beforehand. It wasn't a quick experiment someone knocked up in the lab and put into some humans by any means.
So was there just waiting for a trigger?
Yes. It was being used in other diseases. So the work had already been done. Many years had been spent developing that.
When the research is in that 10-year gap and you find adverse side effects on
Yeah.
animals or people,
Yeah.
what happens?
Then you try and modify the chemistry. So we identify drugs through high throughput screening or through targeted structure-based design, and then we need to actually sort of tinker with the chemistry, basically. So we take an initial drug that's got an initial response, and then we have to tinker with the chemistry to make it druggable, to make it enter the cells well, to make it robust and targeted, and make sure there are no off-target effects, because that's not what we want. We want the very, very specific effects. So yeah, if you end up with a drug that's got off-target effects, you have to scrap that drug, or you have to try and change the chemistry to make it not have those effects. But a lot of drugs, and that's why there's such a lot of money in this, it costs such a lot of money to take a discovery like what we had through to the clinic, because there's so many iterations of the compounds that are developed, and so many that fail, that don't get to the clinic. And that's good, because if they're not safe, we don't want them in the clinic.
Because it seems to me when my mother was in her 90s that the drugs almost seem to be doing more damage than the symptoms.
Yeah, so that's a lot of the problem with a lot of chemotherapeutics for cancer, is that they target cells that divide. So there's a lot of off-target effects that happen to cells. And that's what really cancer researchers are trying to do at the moment, is make much more targeted and much more specific and personalized treatments that don't have side effects, that are really not good to live with.
So do you reach a point with the side effects or having a bad reaction on people where it's acceptable, it's going to cure or retard?
Yeah, so I mean, it will depend on the aggressiveness and the need for the cancer. So maybe some incredibly aggressive cancers the side effects will be more tolerated, and maybe the treatment will be shorter. But ideally, you want to try and stop as many side effects as possible, and that will be really down to the clinician's work to work out exactly the boundaries. But certainly a lot of drugs are terminated in clinical trials because of side effects.
And is there always side effects? Does every action have a reaction somewhere?
I mean, I imagine most drugs have some side effects, but and a lot of these treatments, when you're treating an aggressive growth of cells, are going to have side effects. But the idea is to balance that with the treatment and how necessary the treatment is and how specific it is.
That's it. That's all from me.
Do you want to try and get the analogy again?
Oh, I'm going to try and think of something better for that.
We can cut for a second.
Okay.
I haven't got a slate, but I have got gloves.
Okay, and could that kind of set?
Can we try that?
So the DNA in your cells is generic. All your cells have the same DNA. But what's different is the proteins that are expressed. These are the proteins are what does the job.
Southern. I think the normal term is Hugerhunftemov.
Yeah. So the DNA in your cells is generic. It's the same in all your cells. But the proteins that are expressed in each cell are different. Your eye cell will have different proteins expressed in it than your skin cell. And this is because the proteins are what does the jobs. Other proteins are what do the job.
No, try that again. That's when you can slow down. And it's,
Do I have to go right from the beginning?
yeah.
Oh.
Sorry about that. You have to do the experiment all over.
Yes, that happens a lot. So the DNA in every cell in your body is the same. It's generic for all cells. But the proteins that are expressed are different in each cell. So an eye cell will have different proteins expressed in it from a skin cell. And this is because the proteins are what do the jobs of the cell. They actually action the actions of the genes.
And the cells will be different in different parts of your body.
Yes.
Just say that and carry on.
The cells will be different the cells will be different in the different parts of your body because they do different jobs. So skin cells are different from lung cells, are different from hair cells, but they have the same DNA,
Proteins.
different proteins.
And how do the proteins react with this? That's what we're trying to get to, the proteins. Okay.
How do the proteins react? The proteins are expressed according to the job of the cell because they action what the cell needs to do. They make the cell do those jobs.
tell you what the analogy in my head was going through, which is pulling up and getting some Mexicans. What are you doing gardening today?
Yeah.
It's you get up.
I don't think I can use that analogy.
Get in the back. Come on, take it. What's your job? Build a wall. No, today you're doing the gardening. And it's like, that's, I'm not saying my analogy was
So the
correct, that's what
proteins are
popped
the
into my
workers.
mind, though.
Yeah.
The proteins are the workers.
The proteins
That's
are the
right.
workers. Yeah.
Let's start with that.
Okay.
And so I wasn't so far off, but you pull
Yeah.
up at the truck and get
Okay.
the Mexicans in the back. Don't say that. but
No,
say-
obviously.
take the workers off and they
Okay.
do the job that's given to them.
Yep. So the DNA is the instructions about what needs to be done in the cell, and the proteins are the workers. They're the people, not people. This is going well.
You've got away with guys, even. With the people now.
Okay.
Okay.
Yep, I'm good. So the DNA in the cells is the instructions for what the cell needs to do. And the DNA in each cell in your body is the same. It's generic for all your cells. But the proteins are actually the workers. This is what actually causes the cell to do its job. I think that's the best you're going to get.
Yeah, you're really close.
The DNA, so the DNA is the instructions, but if the DNA is the same in every cell, they live different cells,
Yeah.
do different jobs. How does the
Okay.
DNA know what job is for that particular cell?
Ah,
If
yeah,
the
yeah.
DNA is all the same.
Yeah.
Who's the foreman?
RNA.
Oh. What are you excited?
I knew we were going to get to RNA. I knew it.
Yep.
Waiting there in the wings.
Right.
Okay, well, we're going with the cut rate working.
I'm
analogy.
never going to get this right. It's just going to be like one of these blocks,
Well,
isn't
but
it?
is it true? I mean, I love the fact that we're onto something. The
Yeah.
proteins are the workers.
Yep.
So you've got the ground plan, then you've got, I guess, the architect, if you've got then the builders, then the labourers.
Yep.
Is that a good analogy
Yeah.
or not?
Yeah.
So
Yeah.
let's try it. Before I blow this glove up.
Okay. So the DNA in your cells is generic. It's the same in every single one of your cells. But the DNA encodes the instructions. The RNA is what reads the instructions that's specific for that cell. And then the proteins are the workhorses. So these are the workers that do the jobs of the cell. I think I need to do it. I'm going to do it.
Nailed it.
Well done. Well done.
Cut. Good. Yes.